So my name's Adam Hughes. I'm a student from the George Washington University, and I probably have no business being on a project like this, but the biology department recruited me to help them do some exploratory work on the purple sea urchin, and so that's what I'm here to share with you today. This is very much an exploration in progress. I'm by no means an expert on anything that I say, and I'm really amenable to any criticisms, remarks, suggestions, everything that you can provide. So the background motivation behind this work came from my advisor in the biology department. The purple sea urchin, or the Strongylocentrotus purpuratus, it's a very important model invertebrate. This is like fruit flies and worms, which are other model invertebrates, but it's been sequenced relatively more recently, and it shares over 7,000 genes with us. And the sea urchin, from an immunological point of view, has a very interesting immune system. So just a crude overview on immune systems. Complex vertebrates like us have a two-component immune system. That is, we have an innate component and an adaptive component. The innate component is sort of like an emergency response team. Whenever a pathogen is introduced into the system, the innate immune system executes a general protocol, which might be putting up physical barriers, changing the chemical environment in the organism, and doing things that are going to be like damage control. It's sort of like FEMA, except it works pretty well. The adaptive immune system is a component that's only in more complex vertebrates like us, and it is really the laboratory that figures out what's attacking the body and what are we going to do about it. The adaptive immune system deploys the antibodies that we usually think of that will target a pathogen specifically and keep it from ever coming back and hurting us again. Now, why is this interesting? Well, the sea urchin only has the innate component, the simple component, yet the sea urchin has been around for a long time, and it's in an environment at the bottom of the ocean where it feeds on carcasses and just strains all this dead life through it. And despite all of this exposure to pathogens, the purple sea urchin has a highly robust immune system. So there's a lot going on in the innate immune system that we'd really like to understand. I won't go into the details, but my advisor on this project has determined that one of the mechanisms of the innate immune system to defend itself is to employ groups of these proteins that express this C-type lectin domain. And so the study that we're working on is an exploration of proteins that indeed have this domain. So just to clarify what I mean by a domain, if you look at a protein structurally, you can subdivide it into functional regions. And when these functional regions are identified, we usually refer to them as domains. So you can see them color-coded here. Another way to look at the domains is on this point of view. So if I were to lay this out flat, the sequence region is in gray, and anywhere there's a domain of interest, it's marked with a nice little cartoon. So just like you can do alignments at the sequence level, you can also look at alignments from the perspective of the domains, and it kind of makes it a little bit simpler. It's crude, but there's a lot of reasons to do it. One of the reasons is that domains by themselves are functional. So like we're doing here, we can identify interesting subsets of proteomes to study because they contain a particular domain. Domains are also less prone to mutate over evolutionary timescales because since they have a function, you can't really mutate and have that function be destroyed. Evolution will wipe them out. So they tend to survive longer, kind of unscathed. And also what I'll show you later is that alignments based on domains have more information contained on them than just sequence alignments. You can think about the evolution as the evolution of a language. It's like an evolution of a protein. You can see in this other picture, I have a sentence in Old English evolving to New English through Middle English. And that's kind of similar to what goes on with evolution in proteins, where the domains are like words. Even though the letters change, they don't lose their functionality as quickly. The research goal that I had in mind when I started this was simply, what is the function of the proteins in the sea urchin that have these C-lectin domains? But with on my path of discovery, if you want to call it that, I came up with other research goals that are more general to methods involving the study of these domains. So one thing that's really lacking is general tools to work with domain data from the resource that I use. I know there's some data in other databases that have nice tools, and I'll talk about that. And also, I think there's a lot more work to be done on visualization and analysis techniques just in regard to how we interpret domain data. How do I get my domain data? Well, I use NCBI's Conserves Domains database. There are several databases out there, and I bet a lot of people can make arguments for why this one isn't the best one. The reason I use it is because it's like a domain database aggregator. It pulls in information from other databases out there and some other information from computational algorithms, as far as I understand. And then when you download it, it comes in a nice CSV type, so it's easy to work with. Here's the graphical output from their web tool. So if I put in a query sequence of like 1900 peptides, this is what it's going to return to me. It's going to say this is a domain here, 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 and here. So at the end of the day, this is all that I work with. These are some interesting motifs and regions that have been found by some computational algorithms. And I'm not going to explain this, but this has to do with some manual curation. If you get lucky, you might find some domains that have been aligned by structural biologists, and those are really good domains. But at the end of the day, I just get those domains, and then I perform a whole bunch of really, really bad analysis. The reason I say that, this was exploratory, so when I finally got to where I wanted to be, I looked back on it, and I realized I did everything the wrong way. Everywhere there's purple here, I used a lot of data munging and parsing and just, do it as you go crap that you never want to reuse. And anywhere you see these computers, networks, nodes, is when I used external tools. So the lesson that I learned when I look back and before I publish this is that you need to make sure, and you guys know this, but I learned the hard way, you have to handle your data right before you start working with it. So if you take a protein sequence, you can use BioPython, I learned recently, and their sequence container to do most of the analysis you'd ever want to do in terms of parsing, blasting with that. And I really think, so what I want to do, and what I've done is I've created a little container data type to handle the output of that NCBI tool. And when you have these working together, you have the domain data and you have the sequence data hand in hand. They come together really nicely to do domain-based phylogenetics, visualizations that I'm going to show you, and also to explore domains from a network perspective. A lot of people are working on this, and I think it would be interesting to do it for this CDD database as well. I'll quickly touch on that because I know it's been an interesting topic here. So if I look at all the domains as nodes and their tandem counterparts in the protein, their adjacent nodes as the connections, you can look at the subset of our proteins, for example, as a network where the C-lectin I've just put in the middle and all the other connections as, obviously, as the other nodes. So I don't know much about network exploration, but one question that's interesting is you can ask, in this subset of proteins that I'm studying, are there more C-lectins than I would expect from the entire sea urchin if I had used that entire population, or if I used the entire eukaryote kingdom? And maybe someone that's good with networks can tell me how I would handle that, but I know you can formulate that mathematically using a hypergeometric distribution. So if anyone's ever done networks where you take a subset and you're looking for enrichment or you're surprised in finding something in that network relative to the overall network, my friend Christian Castillo-Davis has a free tool called Gene Merge that literally answers, it solves this for you and says, yes, you should be surprised that you're seeing this, or no, you shouldn't be surprised. I also do crude visualization with Traits UI. I would show you the program, but my VGA out died, so I'm using the last presenter's computer, so thank you for that. But I figured something bad would happen, so I put some slides of it in. So this is the program that I use. It's really crude. And what we have is, these are my unknown proteins in the sea urchin that I'm trying to understand what they do. And sorry for the font, what I have over here is all of the name results from the blast matches. And over here, I have all the domain architectures to these blast hits. So if you take if you do what we did and you take your undergrad and you say, go ahead and blast this and tell me what it is, what they're going to do is they're going to get these names, and they're either going to give you the top hit because it has the best E value, or they're going to just use some human bias and they're going to say, it kind of looks like this one because I like this name and it keeps popping up. Now what we're seeing, oh man, I took a screenshot of the wrong one. Let's look at this one. So this is a pretty common thing that I see. This is my query domains, and I've color-coded them. If you use the PyQt backend in Traits UI, it's easy to get these colors. So if you look at the top blast hit, there's pretty good overlap with your query and your domain, and your top hit. And the majority of the other blasts are dominated by these promiscuous domains. Now, if I could show you the program, I'd be able to show you this better. But these are all scattered around, and it's interesting to see the majority of the blast results tend to have some of these. They're dominated by these promiscuous domains, or they're dominated with hits that are obviously false hits. But if you look at the overlap in the sequence, you would see a lot of sequence overlap here. But you wouldn't know that these are domains, so it's hard to really tell, is that sequence overlap significant or it's not. I can tell you it's not because from the analysis, I know that this is a promiscuous cub domain. It would be easier to show you some more examples of this if the program worked, but sorry that I can't. No. The information on the sequence isn't on this picture, unfortunately. Better tools would have that. But you can usually get a sense that the longer domain architectures are much larger sequences. And that's something I'd like to have if I had that data type working the way I want it to. So let me see, do I have any time left? Yeah. There are efforts to look at the domain architecture, the alignments, by skipping blast completely. So instead of doing the blast step and then seeing what the domain architectures look like in my results, I just look at the alignments from a domain perspective to begin with. And then you have to come up with some sort of scoring mechanism, some sort of metric. And so what we're doing is we're using this tool in conjunction with the one I just showed you. And this is the weighted domain architecture tool. You can use it online. It's a great resource, except that you can't download the data into some sort of nicely parsable format. So you're stuck with the HTML. But for this domain, you can see in the entire eukaryote kingdom, these are the most likely matches just from a domain point of view. This is only using the PFAM database, so there's not a one-to-one mapping between the databases, but it's a really good tool. So at the end of the day, we've come up with some heuristic ways to quantify the data that you see in this set and the data that I showed you, so I can make some general remarks about the systems. So let me just summarize by saying that there's a lot more to explore with domain architectures, and they do kind of provide some really good complementary data when you don't have time to do structural alignments, which are supposedly the best way to find protein homologs. I'd really like to do some work so that anybody can take these CD domain files and do their own exploratory analysis. I was really inspired by some of the talks on PyTables and Network X to actually do this instead of just leaving this as a side project. Because once you use those tools, I think it'll be really easy to access the domain architecture of an entire kingdom. And I think that's where the work needs to go from here. Sorry, kind of threw me for a loop that my computer didn't work. I would like to show you the programs, but you guys have any questions? Does anybody here work with BioPython? So I know you have a PFAM some stuff that works with PFAM, right? Is that sort of like the idea to take the entire PFAM database and like yeah, I saw that, but I wasn't sure. All right, guys. Well, I appreciate your time and listening. Thanks.